Welcome to This Explainer. All right, we are jumping right into something truly mind-bending today. In July 2026, a team at the University of Minnesota basically did the impossible. We're going to unpack how they literally shattered a biological rule that's been running the show on Earth for 4 billion years. We are talking about building life, and I mean quite literally from scratch. Let's get right into it. So, for the entire history of our planet, biology has been governed by one unbreakable rule. Life comes from life. Every single cell that has ever existed in you, me, or anything else came from another cell dividing right before it. It forms this massive, unbroken chain stretching back 4 billion years. And that chain? It has never been broken. Not even once. For all those billions of years, that cosmic rule was perfectly intact. But on July 1st, 2026, that unbroken chain finally snapped. Inside a laboratory, a team of researchers successfully built a cell from entirely dead raw chemical components. And the craziest part is, this wasn't just some static model. This artificial creation can actually feed, replicate, and divide all on its own. Part 1. The 30-year quest. Now, scientists and alchemists have dreamed about pulling this off for centuries, right? But modern synthetic biologists have spent the last 30 years systematically grinding away trying to assemble self-replicating chemistry. To really understand the sheer magnitude of this, Jack Szostak, who is a huge name in origins of life research, pointed out that previous attempts had only ever gotten isolated pieces right. They'd never managed to put it all together simultaneously into one autonomous package. Well, until now. Severin Otto over at the University of Groningen really hit the nail on the head here. As he highlights, lead researcher Katerina Adamala and her team have finally reached the absolute holy grail of synthetic biology. They've taken entirely dead components and arranged them so they spontaneously do what a living thing does. Which brings us to part 2, building the SpudCell. So, how do you actually cross a line that stood for 4 billion years? Well, let's zoom in. You basically need 3 fundamental components. First, you need a liposome made of fat molecules to create an outer boundary, basically separating the inside from the chaos outside. Next, you need circular DNA plasmids to act as your internal blueprint. And finally, a protein expression system, which is the actual molecular machinery needed to read those instructions. To actually jumpstart this whole system, the team engineered a highly precise blueprint. We're talking exactly 90,000 base pairs of genetic instruction. This custom genome is spread out across 7 of those plasmids, acting just like an operating system designed to boot up the cell. And when you put all those pieces together, it's kind of like magic. These completely dead chemical parts spontaneously boot up into a complete 4-step life cycle. The SpudCell absorbs liquid nutrients from its environment, processes those materials to grow, copies its entire 90,000 base pair genome, and then physically splits into 2 new SpudCells. It's the fundamental cycle of life, happening right there in a synthetic bubble. Part 3. The shocking twist. Okay, here is where the creation totally surprised the creators and did something that was absolutely not programmed into it. As these cells replicated, random copying errors naturally popped up, and some of those mutations accidentally made certain SpudCells better at competing for nutrients and dividing faster. So what happens? Those cells reproduced more, and their successful treats spread. Guys, this is unprogrammed, spontaneous, natural selection occurring in an artificial system. The chemistry literally started to evolve on its own. Now, despite possessing all these defining features of life, Katerina Adamala insists we hold off on calling it fully alive just yet. It definitely has some limitations. It still relies on a constant external supply of nutrients and ribosomes, it lacks a waste removal system, and it has absolutely 0 defense mechanisms against its environment. So it's a massive step closer to life for sure, but strictly speaking, not quite life itself. Part 4. Past and future. Reaching this milestone after 30 years and billions of dollars shifts our entire paradigm in 2 totally opposite directions. Looking backward, SpudCell acts as this fascinating bridge. It proves exactly how simple chemicals in the oceans of early Earth could have naturally self-organized. But looking forward, it ushers in a radically new era of biotechnology. Looking to the future, this paves the way for a brand new class of designer cells. Imagine artificial cells built from scratch, carrying instructions we wrote, programmed specifically to navigate your bloodstream. They could hunt down cancer, deliver life-saving drugs to highly precise locations, or even produce therapeutic proteins exactly where your body needs them. It's an unprecedented level of control over our own biology. But taking a step back from the medical side, there is a massive philosophical weight to this whole experiment. Joe Buchhoven from the Technical University of Munich put it perfectly. He said, if you want to understand what life is, you need to first build life. By assembling it with our own hands, we are finally testing the very outer boundaries of biology. As we reflect on this single impossible event, where a literal bubble of fat and 7 plasmids turned chemistries into a biology that can feed, divide, and evolve, I want to leave you with one profound thought. If entirely dead components can be arranged to do absolutely everything a living cell does, what actually is life? Think about that. Thank you for joining me on this explainer, and keep questioning the amazing world around us.